Toxoplasma gondii is a microscopic, single-celled parasite that currently resides in the tissues of approximately 1/3 of the global human population. Despite its massive reach, it faces a rigid biological constraint. The parasite can only complete its life cycle and undergo sexual reproduction within the intestinal tract of a feline. This creates a daunting evolutionary obstacle. Because its eggs are shed in cat feces, they are typically ingested by foraging rodents. To return to its definitive host, the parasite must ensure that its intermediate host, a rat, is eaten by a cat. However, rats possess an innate, life-saving aversion to the scent of felines. If the rat successfully avoids the predator, the parasite reaches a dead end. To survive, Toxoplasma has evolved to actively override the rodent's survival instincts. Following infection, the rat's behavior shifts. It loses its natural fear of cat odors and, in many cases, develops a dangerous attraction to them, lingering in areas where predators have marked their territory. This behavioral hack results from physical infiltration. The parasite targets and insists within the amygdala, the brain's primary center for processing fear and threat. This is a surgical, localized intervention. The rat's motor skills, general intelligence, and other cognitive functions remain intact, ensuring it remains a mobile, active participant in seeking out the predator. By stripping away only the fear of its predator, the parasite successfully turns the host into a willing delivery vehicle to complete its own life cycle. To understand how a single-celled organism can steer a complex mammal, we have to look at the chemical engineering happening inside the infected brain. Toxoplasma possesses two specific genes that allow it to manufacture its own supply of an enzyme called tyrosine hydroxylase. This chart shows dopamine synthesis. Tyrosine hydroxylase is the rate-limiting trigger, the bottleneck determining dopamine production capacity. By introducing its own enzyme, the parasite creates a localized dopamine flood. This reconfigures neural pathways, overloading receptors so cat odors trigger reward instead of fear. The parasite does not simply damage the brain, it weaponizes the host's primary motivation chemical against its own survival. Roughly 2.5 billion people, 1/3 of the human population, currently carry a latent Toxoplasma infection. Because modern humans are rarely preyed upon by felines, the parasite is trapped within our bodies. It has reached a biological dead end with no way to return to its feline host. Despite being trapped, the parasite's autonomous dopamine factory does not shut down. It continues to operate inside human brain tissue indefinitely. This unintended, continuous chemical activity creates a systemic disruption in human baseline behavior. In humans, this chemical disruption correlates with a broad behavioral shift toward increased impulsivity and an elevated tolerance for risk. This chart links latent infection to behavior. Infected individuals are significantly more likely to be in traffic accidents. This risk appetite appears professionally, showing a 1.8 times spike in entrepreneurial activity. Most severely, psychiatric data reveals a 2.7 times greater risk of developing schizophrenia. What evolved as a targeted survival mechanism for a rat acts as a chaotic, blunt force neurological disruptor in humans. This evidence invites us to reconsider how we define our own personality traits and the decisions we make every day. We generally operate under the assumption of absolute conscious free will, yet Toxoplasma demonstrates how easily our internal narratives can be swayed by subtle neurochemical shifts. Core aspects of our identity, from our appetite for risk to our baseline levels of impulsivity, rely on chemical thresholds that are remarkably susceptible to outside interference. It suggests an unsettling possibility that what we feel as a conscious choice might sometimes be a post-rationalization for a chemical imbalance. A microscopic, single-celled organism has developed the capacity to fundamentally overwrite mammalian behavior to serve its own reproductive needs. And despite our complex cognition, our brains run on the exact same hackable chemical hardware as the rodents the parasite first evolved to control. This implies that some of our deepest personal urges and life-altering decisions might not actually belong to us. Free will becomes a difficult concept to defend when the levers of our minds are being silently pulled by a parasite.